This one, before the program, all I've done is I've taken some of those leaves that I collected this morning and I've mashed them up. You can see that I have kind of a leaves, kind of smashed up substance in here. I've ripped up some leaves and I've smashed them up and soaked them in just a little bit of isopropyl alcohol. What that's done is it's actually let me release some of those beautiful pigments that we find inside the leaves. So this one is a pretty bright green color because I got it from these very, very green maple leaves that are pretty early on in their process of breaking down. But then I also took some of those red sugar maple leaves. These ones are going through that color change that I'm sure we all recognize is going on right now outside with most of the leaves that we see. I've done the same thing, and you can see that this substance is a little bit more red. What we're going to be teaching people how to do today is how you can actually separate the different types of pigments that we find inside leaves. The green is what we call chlorophyll, and this is definitely the most prominent color that we see in leaves for most of the year. Chlorophyll is green. It comes from those chloroplasts, which we find inside most plant cells or all plant cells, and gives them that bright green color. And we're going to use a process called chromatography in order to separate the pigments that we have down there inside our liquid. I've done a little bit of pre-work before our time together here. I've just taken a small strip of paper towel, but a coffee filter would work great for this as well. I've kind of taped it to a skewer, or you could use a popsicle stick, and I'm just going to set it down just barely inside our liquid, and you can see that that liquid is starting to slowly travel up the strip of paper towel. The process of that liquid traveling up the paper towel is called capillary action. And that's actually the exact same action that plants use in order to draw water out of the ground and get it into their various leaves and branches in order for them to survive. So I've done this twice. So we have one that we're dipping down here inside our green pigment, that chlorophyll, but we're also going to set one down inside our red liquid. Now, this process takes a little bit of time. Capillary action works slowly, but that's a good thing. This process that we call chromatography is going to actually allow us to separate the different pigments that we find inside leaves. The color that we see in red leaves is because of a chemical called anthocyanin. Anthocyanin only appears as chlorophyll, the green pigment, starts to break down. And this happens because these trees start to get less sunlight as we move into fall. Now, you might know that just last week, we actually had our first official day of fall. On the 22nd, we had the autumnal equinox, which means that from this point on until the winter solstice, our days are getting shorter and shorter and shorter, meaning we are getting less sunlight, which means less of that photosynthesis is happening inside these leaves. And that's really what's responsible for this color change. As they get less sunlight, there is less chlorophyll being produced by the process of photosynthesis. These trees go into winter mode, and they start to turn red as the chlorophyll breaks down and turns to anthocyanin. But of course, we see other colors besides just red and green. I have some kind of yellow leaves here, and that's because of a pigment called xanthophyll. And xanthophyll is actually present all year round. It's always in the leaves. We just don't notice it until the chlorophyll starts to disappear. But our process of chromatography, which is happening down on our strips of paper, is going to allow us to separate all the pigments that could possibly be inside the leaf right now. I set up a couple before our program here today, and you can see that on this one, if I zoom in maybe even just a little bit further, we see green, which is that really well-known chlorophyll right here, this band of green pigment, but we can also see a line of yellow, which is that xanthophyll pigment. So again, this is one of those really cool activities that people can go out and try at home. It doesn't require any sort of expensive equipment or dangerous chemicals. All you really need are leaves, various colors, various stages of decomposition, some green, some red, maybe some yellow if you can find them, a couple strips of paper towel, some isopropyl alcohol, and a cup. It's a really, really easy experiment that you can do at home in order to help separate these pigments and identify a process that's going on out in nature all the time. Things that we might not notice, but are really important for the ecosystem around us. So we do experiments like this all the time on Echo Live. We've got more to talk about today. We'll dive in a little bit deeper about how you can identify the species of trees that you see in your neighborhood or when you go on a walk. Maybe not today because I do think it's supposed to rain, but maybe tomorrow. But again, people can tune into our experiments every Wednesday at 2:30 p.m. on our Facebook page, our YouTube channel, and right here in Zoom.